We previously saw an example of how E. coli can manipulate, how it can regulate the expression of its gene using an inducer. Now let's look at another example of how genes are regulated using repressor. We are going to look at another type of operon, the trip operon. The principle is the same. We have structural genes. These are the structural genes which, when translated, result in help or assist the cell in making tryptophan. Upstream from this structural genes is the same as you saw in the lac operon. There is a promoter and an operator. We know what is the function of both these sequences. The operator binds the repressor. When repressor is bound to operator, DNA cannot be transcribed. So when there is no tryptophan, the cell needs to make tryptophan. The repressor will be inactive. It will not be able to bind operator. RNA polymerase will be free to bind the promoter region, and this would result in production of the proteins necessary for the biosynthetic pathway for tryptophan. Now let's see what happens if tryptophan is here. Remember, that was the previous system. We had an inducer. Here we have a repressor. And the repressor needs a co-repressor to bind operator. The co-repressor is the end product of this metabolic pathway, the tryptophan. So if the cell already has a lot of tryptophan in the system, cell already has a lot of tryptophan available to construct proteins, there is no need for the cell to keep on making more tryptophan. It will be a waste of energy and resources. So when tryptophan is present, it acts as a co-repressor. It binds the repressor. When it binds the repressor can go through a conformational change. The domain of this protein becomes available to bind the operator. The DNA binding domain becomes available. It prevents the RNA polymerase from binding the promoter and conducting the messenger RNA, formation of messenger RNA. It prevents that. So here we have a system. Both the systems we have talked about are negative control. The regulatory molecule is the repressor, is preventing transcription. So the trip operon and the lac operon are the negative control. We'll look at an example of positive control. So let's do that. Here, the regulatory molecule will, when it binds the specific region of DNA, it will have a positive control, meaning that it will facilitate transcription rather than hinder it. So let's see an example. What if bacteria has some glucose but very low glucose, and it has abundant lactose? When glucose levels are low, it results in production of a molecule called CAMP. It is cyclic adenosine monophosphate. We already know what ATP is, so we also know what AMP is. Cyclic AMP or CAMP is a compound derived from adenosine monophosphate. This compound plays a similar role in humans also. When we are hungry, when this compound is produced, we feel hunger and desire to eat. It has similar function in bacteria. Let's look at that. When cyclic AMP is present, when low glucose concentration, and we have lactose in the system, so the repressor cannot bind the operator, cyclic AMP binds a protein called CRP, which basically stands for receptor protein, which is a receptor protein, cyclic AMP receptor protein. When cyclic AMP binds it, this protein, this complex, rather than binding the operator, it binds the promoter, and it facilitates the docking of RNA polymerase at this site, and resulting in production of the structural genes, which are the same genes which we saw in the lac operon. So this is what happens when glucose is low and lactose is abundant. What happens is if the glucose is high, then this molecule CRP will not be able to bind promoter. RNA polymerase will bind the lactose, the lac operon promoter, but now the efficiency is very low. When the efficiency is very low, very few molecules of these structural genes will be produced. So under conditions when glucose is present, although even if lactose is available, very few structural genes will be transcribed and translated, which play a role in metabolism for lactose. So we have looked at 3 examples, 2 negative control examples, 1 positive control example. The 2 negative control examples were number 1, the lac operon with the inducer in it, the inducible system. The other was repressible system, the trip operon, and the positive control where we saw how an operon or how promoter's ability to recruit RNA polymerase can be enhanced by a protein that is sort of acting as an activator. So we have seen 3 examples how a small bacteria can regulate expression of its proteins depending upon its needs or the environmental changes.